nominating these people, because without nomination, we wouldn't be able to select the winner. And it's with great pleasure that this year we will award the awards, the plaque and the money as it did it tonight, but it's with great pleasure that I now ask Jeff Tam to come up and introduce the 2014 Carl W. Gottschalk Distinguished Lectureship winner, Susan Wall from Emory University. Jeff.
Thanks, Edwin. And it is really an honor and a privilege to get to introduce my good friend and colleague, Dr. Susan Wall, who is the 2014 Carl W. Gottschalk Distinguished Lecturer of the APS Renal Section. Susan, like Dr. Gottschalk, is a professor of medicine and physiology, only she's at Emory and not at UNC. Susan's training, she had a BS in chemistry from the University of Washington, received her MD from St. Louis University School of Medicine. She did her internal medicine and nephrology clinical training at UCLA, and was both a research fellow at UCLA and also at the LKEM in Bethesda, which is when I first met Susan, while we were both fellows at the time in Dr. Mark Nepper's lab. Susan has held 2 faculty positions. After the LKEM, she went to the University of Texas in Houston, where she rose from assistant professor up through the ranks until in 2003, when she was actually the first faculty member I was able to recruit to Emory after I had become division director. Probably my best individual lupus division director was convincing Susan to come to Emory, and it's been great fun working with her over the past decade. Susan has been very active in many ways. Highlighted here is her APS service. She served as renal section treasurer, and then as renal section chair. She was an associate editor of AJP Renal for 12 years, and this year joined the APS Publications Committee. You'll hear a lot about her research, of course, in the subsequent lecture, so I'm really not going to talk much about it, except to highlight the 2 foci of proton and bicarbonate transport along the collecting duct, and the role of chloride transport in the cortical collecting duct in the regulation of blood pressure, which is the topic she'll talk about today. But what I want to share with you is that Susan is the only person I know who has actually been on all 7 continents. She's incredibly well traveled, and shown here, you can see each of these red dots represents a place in the world other than the continental U.S. Where Susan has visited. And of course, this being a scientific lecture, you're going to want some data to back up this claim, so I have it for you. Here's Susan in Alaska. So she's been almost to the top of the world. And she's also been in Antarctica, so she's been at the bottom of the world. The weather looks a little worse, actually, in Antarctica than it did in Alaska in these 2 pictures. She's been to Machu Picchu, although I'm not sure we can actually make her out in this picture, but it looks like a very beautiful location. And she's been in Angkor Wat in Cambodia. She's been to Italy. And here she is in front of the Great Pyramid of Giza in Egypt. And here she is in front of the famous Opera House in Sydney, Australia. So as you can see, she really has been all over the world, all the continents. But the last 2 pictures I'll show you actually have a little story that go with them. This is Susan on a trek up the mountain of K2, which is on the China-Pakistan border. And for those like me who didn't know it, K2 is actually the 2nd tallest mountain in the world. And this is approaching it from the north side or from the China side of the mountain. And to get there, you actually have to ford quite a few rivers. And to do that, you have to do it on camels, because the rivers are fairly deep. And shown here is part of that trek. And in this picture, Susan has actually fallen off the camel. They did not tie the stuff onto the camel very well. I guess they didn't do it right, so the gear fell off, and Susan with it. And she said that's happened to many individuals, although she believes she's the first Western woman to ever fall off a camel going to K2. But I think this is an analogy for today's world of science, where many of us fall off the camel, so to speak, as our grants or our papers get less than the comments we would like. But Susan got back on the camel and continued on and continued her trek to K2. And I think this is a testimony both to her and the magnificent places she's visited, but it's also an allegory for how we need to do science. We may fall off the camel, but we got to get right back on and keep going. And so, without further ado, it's my pleasure to introduce Dr. Susan Wall as this year's Karl Gottschalk awardee. Susan?
Well, thank you, Jeff, and sorry for that little technical distraction. This award is a tremendous honor, and of course I have many people that I want to thank for it, including the American Physiological Society, and then of course I want to thank the mentors that I had as a postdoctoral fellow where I got my start, and in particular I was in the labs of Jeff Kraus and Shlomo Wallem at UCLA, and then more recently Mark Nepper at the NIH. Then when I was a faculty member, I had the opportunity to have some really very supportive division chiefs, including Tom DeVos and Bruce Cohn at the University of Texas, and now my current mentor and division chief, Jeff Fan. And then, of course, most of the work that I'll present was not done by me, but was rather done by all of my collaborators, most of which are listed on this slide. And so I'm very grateful for their participation. And finally, I want to thank my brother, who's here to celebrate with me and also to give me moral support. So what I'm going to be talking about today is the interaction of Pendrin and the epithelial sodium channel in the pressor response to aldosterone. Well, Pendrin syndrome, which we now know is due to inactivation mutations in the gene SLC26A4, was originally described by Dr. Von Pendrin more than 100 years ago. And what Dr. Pendrin observed was a family with 10 children. And of these 10 children, 2 had goiter and were deaf. Since that time, there have been more than 100 disease-producing mutations that have been described. We know that the inheritance is autosomal recessive. However, no renal phenotype has been observed. What brings these people into the doctor is not their kidney problems, but rather their hearing loss. Although occasionally they have goiter in about a third of the patients, and occasionally the patients are hypothyroid. Then about 100 years later, Lorraine Everett cloned the gene responsible for Pendrin syndrome by positional cloning. At the time, she was a postdoctoral fellow in the lab of Eric Green. Well, the gene responsible for Pendrin syndrome we now call SLC26A4, and Pendrin is referred to as the gene product of SLC26A4. Well, after Lorraine cloned the gene responsible for Pendrin syndrome, she developed a Pendrin knockout mouse, and that mouse recapitulated the deafness that we see in Pendrin syndrome, although the mouse has no thyroid phenotype. But after the gene was cloned, there were 2 questions that we could immediately ask. The sequence of Pendrin predicted the sulfate transporter, but we were better able to measure directly what Pendrin's transport characteristics are, in addition to determining where it's expressed and how it's regulated. Well, initially, Larry Karnitsky and later Manucha Soleimani expressed Pendrin in heterologous expression systems and determined that it's an electroneutral, sodium-independent chloride bicarbonate exchanger. Well, Ines Rayo then, too, was a postdoc in Eric Green's lab and was given the job of determining where Pendrin is expressed throughout the body. And of course, he found that it's highly expressed in ear and also in thyroid, which was no surprise. But what was surprising was that it was highly expressed in kidney, and in particular in the aldosterone-sensitive region of the nephron, or the distal convoluted tubule, the connecting tubule, and the cortical collecting duct. So Ines did immunofluorescence studies of the cortical collecting duct to determine where Pendrin is expressed. And the micrograph is shown in this slide. So what she found is that Pendrin, which labels red in this microgram, is expressed in the apical region of a minority cell type within the cortical collecting duct. Well, of course, Innes and Eric were in the Human Genome Institute, so they're not used to thinking about cortical collecting duct function on a daily basis. So they showed the data to Mark Knepper, who immediately recognized that Pendrin is expressed within intercalated cells. And this gave us tremendous clues in terms of what Pendrin is probably doing in kidneys. And how these clues became apparent are shown in the next series of slides. Well, in the cortical collecting duct, there are two cell types. There are principal cells and there are intercalated cells. Principal cells express the epithelial sodium channel on the apical plasma membrane that mediates the absorption of sodium. That then generates a lumen negative potential, which provides the driving forces for the secretion of the absorption of ions such as potassium. The minority cell type are intercalated cells, and intercalated cells have three subtypes: type A, type B, and non-A, non-B intercalated cells. Type A intercalated cells express the proton ATPase on the apical plasma membrane, which is greatly upregulated in models of metabolic acidosis. The result is increased secretion of proton equivalents and correction of the acidosis. Type B intercalated cells express a sodium-independent chloride bicarbonate exchanger on the apical membrane, which is greatly upregulated in models of metabolic alkalosis, such as the administration of aldosterone. Non-A, non-B intercalated cells also express this putative apical sodium-independent chloride bicarbonate exchanger, although this cell type is not as well understood. So since Pendrin and the sodium-independent chloride bicarbonate exchanger co-localize to the same region within the same cells, Mark Knepper proposed that Pendrin may be responsible for this putative sodium-independent chloride bicarbonate exchange process. So Mark called me one day and he suggested that I measure bicarbonate secretion in the Pendrin knockout mouse, which I did. So the question I asked is whether or not Pendrin gene ablation modulates the secretion of bicarbonate and the absorption of chloride in the cortical collecting duct. So to do that, I treated wild-type and Pendrin knockout mice with an aldosterone analog and measured chloride absorption and bicarbonate secretion in their CCDs when perfused in vitro. This slide shows bicarbonate and chloride flux in a CCD from a wild-type mouse that has been treated with dope. And as you can see, this CCD secretes bicarbonate and has absorbed chloride, as you'd expect based on a number of studies that were done about 30 years ago. However, in contrast, CCDs from the Pendrin knockout mice treated identically did not secrete bicarbonate and they did not absorb chloride. So I concluded that B-cell apical chloride bicarbonate exchange is completely dependent upon the expression of Pendrin. Well, since it was known by these older studies that apical chloride bicarbonate exchange is extremely sensitive to aldosterone, Jill Berlander and I asked whether or not Pendrin expression is also upregulated by aldosterone. So to do that, I collaborated with Jill at the University of Florida, and she looked at Pendrin abundance and subcellular distribution using immunogold cytochemistry and morphometric analysis. This is a slide, an electron micrograph of a type B intercalated cell that has been labeled for Pendrin. And antigen-antibody interactions are detected by colloidal gold, so they appear as black dots. So there are two prominent characteristics of this B cell. First of all, it has a relatively smooth apical plasma membrane. And the second thing is there are relatively few black dots, relatively little Pendrin expression along the apical plasma membrane. Instead, there's abundant Pendrin immunolabels in the subapical space. So this slide shows an electron micrograph of a type B intercalated cell from a deoxycorticosterone treated mouse, which is an aldosterone analog. And you can see that there are two fundamental differences in this cell. First of all, this cell is very prominent apical microvilli. And the second thing is that there's abundant Pendrin immunolabel along the apical plasma membrane. So Jill quantified Pendrin labeled along the apical plasma membrane per cell in type B intercalated cells and non-A, non-B intercalated cells in mice that were given the vehicle or mice that were given this aldosterone analog. You can see the apical plasma membrane Pendrin immunolabel went up in both cell types, but it increased most markedly in the type B intercalated cell, going up about six-fold in response to aldosterone. So as I mentioned, people in mouse models of Pendrin syndrome really have no renal phenotype under basal conditions. They have normal acid-base balance, normal fluid electrolyte balance, and normal renal function. So we ask if a renal phenotype becomes apparent when Pendrin is stimulated, such as with the treatment of aldosterone. So we ask if people or mouse models of Pendrin syndrome have a defect in their ability to regulate the excretion of hydroxyl equivalent or chloride. So in the first set of experiments, we measured arterial blood gases in Pendrin null and wild type mice under basal conditions, or just following a standard rodent diet, which is fairly high in salt. And so as you can see under these basal conditions, arterial pH and also bicarbonate concentration are really similar in wild type and Pendrin knockout mice. However, if you give these mice an aldosterone analog, arterial pH goes up in both groups of mice, although goes up most strikingly in the Pendrin null mice, consistent with a reduced ability to excrete hydroxyl equivalent. So as I mentioned, Pendrin null mice developed an enhanced alkalosis when stimulated, such as with aldosterone, but we and others asked why arterial pH is unchanged in these mice under basal conditions. So the answer to this is probably largely due to compensation along the nephron by other chloride transporters. And in particular, there seems to be an interaction between the thiazide-sensitive sodium chloride cotransporter in the distal convoluted tubule and Pendrin. And in fact, there was a nice talk today by Paul Grimm and Paul Welland's lab that they may interact through alpha-ketoglutarate. Well, Dominique Elidari's lab has shown that if you knock out NCC, you get a dramatic upregulation of Pendrin. So what happens to acid-base balance and fluid and electrolyte balance when you knock out both NCC and Pendrin and study them under basal conditions? So as I mentioned before, we've observed that bicarbonate concentration is similar in wild type and Pendrin knockout mice under basal conditions. However, Manucha Soleimani's group has shown that if you knock out both Pendrin and NCC, you have a dramatic increase in serum bicarbonate concentration. So does this happen in people? In other words, with people with a two-hit like this, with inactivation of Pendrin and NCC, you develop an alkalosis. Well, this was actually reported in a case report about 6 years ago, where they described a person with Pendrin syndrome that received a thiazide diuretic. Well, as you know, thiazides primarily target the sodium chloride cotransporter. So this person was the equivalent of Manucha Soleimani's NCC null, Pendrin null mouse. And this is what happened. Under basal conditions, this person with Pendrin syndrome presumably had normal serum bicarbonate concentration. However, following 2 weeks of a thiazide, this person's serum bicarbonate concentration increased from probably about 25 to about 44 millimolar, which was accompanied by profound chloride wasting and vascular volume contraction. The thiazide was then discontinued, and the person was given KCl infusion, where his vascular volume contraction and his alkalosis promptly returned to normal. So in mouse models of Pendrin syndrome, and possibly in people with Pendrin syndrome, there's an impaired ability to excrete hydroxyl equivalent. So since Pendrin mediates chloride absorption, we asked if Pendrin null mice have a defect in their ability to regulate the excretion of chloride. And to do that, we did balance studies, both on a high and a low salt diet, in wild type and Pendrin knockout mice. And so initially, we put both sets of mice on a diet containing a reasonably high content of sodium chloride, and then measured 24-hour excretion of sodium and chloride. And as you can see, following this high salt diet, the excretion of both of these ions is roughly similar. However, if the diet was then changed to a low salt diet, at least initially, the Pendrin knockout mice, which are the gray bars, excreted more sodium and excreted more chloride than their wild-type littermates. In addition, following sodium chloride restriction, the Pendrin null mice have evidence of intravascular volume contraction. You know, they lose more weight, their CUN and their hematocrit go up, and they have lower blood pressure, I'll describe later. So we asked if Pendrin gene ablation also changes blood pressure. And to do that, I measured blood pressure in Pendrin knockout mice and wild-type littermates under basal conditions or following a high salt diet, and then following the administration of an aldosterone analog. So as you can see, under basal conditions, systolic blood pressure is similar in the wild-type and the Pendrin knockout mice. If, however, the mice are given an aldosterone analog, blood pressure went up markedly in the Pendrin in the wild-type mice, but did not change appreciably in the Pendrin null mice. Dominique Alladari's group has looked conversely at the effect of overexpressing Pendrin on blood pressure. And in particular, he looked at the effect of a high salt diet on blood pressure in wild-type mice and also in mice that overexpress Pendrin. And as you can see, in wild-type mice, if the mice are changed from a normal to a high salt diet, there's very little increase in blood pressure, which is similar to what you'd expect in people. However, in the mice that overexpress Pendrin, there is a dramatic increase in systolic blood pressure that is very sensitive to the intake of chloride. So, Pendrin clearly regulates blood pressure in mice, but does it also regulate blood pressure in people? Well, this has not been completely studied, but there was an interesting study, retrospective chart review that was done by an NIH group, which looked at the incidence of hypertension in people with Pendrin syndrome and also in their unaffected family members. And this slide shows the results. In 21 persons with Pendrin syndrome, there was 2 alleles with inactivation mutations in SLC26A4. They observed no hypertension. However, in their family members that either had no mutant alleles or only had 1 mutant allele, the incidence of hypertension was somewhere between about 9 and 16%. So Pendrin gene ablation may be protective against the development of salt-sensitive hypertension. So the key points are aldosterone regulates Pendrin and also ENaC, abundance and function, which I'll come back to. Pendrin gene ablation blunts the pressure response to aldosterone. And finally, overexpressing Pendrin leads to salt-sensitive hypertension. So then we asked why Pendrin knockout mice excrete more sodium and chloride. Well, since Pendrin is an important mechanism of chloride absorption, at least in the cortical collecting duct, we reasoned that if you knock out Pendrin, you probably lose an important mechanism of chloride absorption along the nephron. However, Pendrin does not mediate the transport of sodium, so the cause of the natriuresis that we observed in these Pendrin null mice was not as apparent. However, in Vladimir Peck measured transepithelial voltage, which really is an index primarily of ENaC-mediated sodium absorption, in Pendrin knockout mice and also wild-type mice that have been treated with furosemide, which increases ENaC and Pendrin. And what he observed is a robust lumen-negative transepithelial voltage in the wild-type mice, which was almost completely obliterated with ENaC inhibitors. In contrast, in the Pendrin null mice, transepithelial voltage was very low and not appreciably changed with the application of the ENaC inhibitor. So how this reflects sodium absorption through ENaC is shown in this slide. ENaC mediates the absorption of sodium. It generates a lumen-negative voltage. And then with the application of the ENaC inhibitor, that voltage is eliminated. And one can measure the change in voltage with benzamyl to get an index of ENaC-mediated sodium absorption. Vladimir then went on to measure sodium absorption directly and cortical collecting ducts from aldosterone-treated wild-type and Pendrin knockout mice. And he observed that sodium absorption was robust in wild-type mice. Sodium absorption was also observed in Pendrin null mice, but it was much lower in magnitude. And the sodium absorption of both of these groups was almost completely eliminated with the application of benzamyl to the luminal fluid. So when Vladimir quantified then sodium absorption, both the total sodium absorption and the benzamyl-sensitive component of sodium absorption, which reflects ENaC-mediated sodium absorption, he observed that both total and the benzamyl-sensitive component of sodium absorption falls by about 50% with Pendrin gene ablation. So our group explored more in detail what might be the molecular mechanism for this interaction of Pendrin and ENaC. Well, ENaC consists of three subunits, the alpha, the beta, and the gamma, each of which is required for full channel activity. So Young-Ki Kim profiled the nephron of wild-type and Pendrin knockout mice, examining the abundance of the major mechanisms of renal sodium transport along the nephron, in particular NHE3, NKCC2, the sodium chloride cotransporter, and the epithelial sodium channel. And what she found is under basal conditions when circulating aldosterone concentration is low, such as following a high-salt diet, sodium transporter abundance, including ENaC, is really very similar in the wild-type and the Pendrin null mice. However, when circulating aldosterone is high, such as following dietary sodium chloride restriction or an aldosterone infusion, ENaC subunit abundance and ENaC function are lower in the Pendrin null than in the wild-type mice. And in particular, she noticed that the abundance of the 70 kilodalton fragment of gamma ENaC, which is the active fragment of gamma ENaC, is much lower in the Pendrin null than in the wild-type mice. So ENaC subunit abundance is reduced in kidneys from Pendrin null mice. Then in further studies that we did with Jill Verlander, we asked whether or not Pendrin gene ablation changes ENaC subcellular distribution. This is a immunohistochemistry that Jill did, showing a cortical collecting duct from an aldosterone-treated wild-type mouse and a Pendrin null mouse that had been labeled for beta ENaC. And as you can see in the CCD from the wild-type mouse, beta ENaC immunolabel is very intense and very discrete along the apical region of this nephron segment. In contrast, in the Pendrin null mice, Pendrin immunolabel is much more diffuse and less discrete in the apical region. So Jill then quantified the apical beta subunit label in the most apical 10% of the principal cells in both groups of mice. And to do that, she quantified the intensity of beta subunit immunolabel in the apical most 10% of the cell and divided that by total beta ENaC immunolabel in that cell. And this is what she found. That the relative expression of the apical 10% of the beta immunolabel is reduced by about 40% with Pendrin gene ablation. So the key point of these studies are that Pendrin gene ablation reduces ENaC subunit total protein abundance and the relative abundance of ENaC in the apical regions of principal cells. So since Pendrin modulates ENaC subcellular distribution, we asked if Pendrin modulates ENaC function when ENaC is constitutively expressed on the plasma membrane. And to do that, we generated, first of all, the next series of slides review a large mechanism of how ENaC abundance and subcellular distribution are regulated. Well, ENaC, of course, is expressed on the apical plasma membrane. In response to aldosterone, the ubiquitin ligase MED42 is dephosphorylated, which then associates with ENaC, causing ENaC to be ubiquitinated. ENaC is then endocytosed and is degraded in proteasome or lysosome. However, in little mice, there is a truncation mutation that occurs in the beta subunits of ENaC. That then results in the loss of this PY motif, which prevents NED42 from associating with this epithelial sodium channel. So once again, in response to aldosterone, NED42 is dephosphorylated, but it cannot associate with ENaC. So the result is a greatly enhanced expression of ENaC along the apical plasma membrane. So we made use of this Little's model to generate a Pendrin null mouse that constitutively expresses ENaC on the apical plasma membrane. So to do so, we bred Pendrin null mice with a mouse model of Little's syndrome to generate mice that are homozygous Pendrin null and homozygous for the Little's mutation. So this slide shows a cortical collecting duct from an aldosterone-treated wild-type and Pendrin null mouse that has been labeled for the gamma subunit of ENaC. And once again, like with the beta subunit, gamma immunolabel is very intense and discrete in the apical region of these nephron segments. However, it's much less intense and less discrete in CCDs from Pendrin null mice. However, when the Little's mutation is introduced, Pendrin gamma ENaC immunolabel is very intense and very discrete in the apical region of mice that Little's mice that carry wild type Pendrin, as well as Little's mice that are Pendrin null. So we asked if Pendrin gene ablation changes ENaC function when ENaC is constitutively expressed on the plasma membrane. And so once again, we measured the change in transepithelial voltage observed with ENaC inhibition, or the benzamyl sensitive complement voltage, as an indicator of ENaC-mediated transport. How this was done, once again, is shown in this slide. ENaC mediates the absorption of sodium, which generates a lumen-negative voltage. With the application of benzamyl, this mechanism is eliminated, and one can measure the benzamyl-sensitive complement voltage as an index of ENaC-mediated sodium absorption. And this is what Vladimir found. She found that in aldosterone-treated mice, Pendrin gene ablation reduced the benzamyl-sensitive complement voltage by about 50%. If the Little's mutation is introduced into the Pendrin null mice, benzamyl-sensitive voltage increases by about twofold. However, the benzamyl-sensitive complement voltage is still lower in Little's mice that are Pendrin null relative to Little's mice that harbor wild-type Pendrin. So we reasoned that constitutively overexpressing ENaC on the plasma membrane does not eliminate Pendrin-dependent changes in ENaC function. So we reasoned that Pendrin gene ablation must reduce ENaC-mediated current through an additional mechanism. And in particular, we hypothesized that Pendrin gene ablation also changes ENaC open probability. So to answer this question, we collaborated with Doug Eaton and Fong Bao, who treated Pendrin knockout in wild-type mice with aldosterone, then dissected CCDs, split them open, and took excised patches of principal cell apical membrane and measured ENaC single-channel reporting. And this is what she found. This slide shows ENaC single-channel reporting from an aldosterone-treated wild-type mouse and also a Pendrin knockout mice. And as you can see in this slide, the channel is open much more in the patches from the wild-type mice than the Pendrin knockout mice. So Fong then quantified channel activity, and she found that Pendrin gene ablation reduced channel activity by about 50%. But since ENaC channel activity is the product of the number of channels times the open probability, she looked at both of these components separately. And what she observed is that with Pendrin gene ablation, the number of channels in the patch went down by about 40%, which is consistent with Jill Berlander's data, showing that Pendrin gene ablation reduces the relative expression of ENaC in the region of the plasma membrane. But in addition, she also observed that Pendrin gene ablation reduces ENaC open probability by about 40%. So the key points are that with Pendrin gene ablation, ENaC function is reduced to to a fall in ENaC subunit protein abundance, due to a fall in ENaC abundance in the apical region relative to the cytoplasm, and also due to a fall in ENaC open probability. This reduced ENaC expression that we found in the Pendrin nullibi contributes to the fall in blood pressure that we observed with Pendrin gene ablation. However, the nature of this interaction between Pendrin and ENaC was not known at that point. So how might Pendrin regulate ENaC abundance and function? Well, we know some ways it could not regulate it. For example, while ENaC and Pendrin both localize to the same nephron segment, they're clearly of different cell types. So Pendrin cannot regulate ENaC through a direct protein-protein interaction. Similarly, we measured the circulating concentration of a number of hormones, including vasopressin, renin, aldosterone, corticosterone, and thyroid hormone, and we found that we could not see that changes in any of the concentrations of these hormones could explain the fall in ENaC abundance and function that we observed with Pendrin gene ablation. And finally, the Pendrin-dependent changes in ENaC abundance and function appear to be fairly kidney-specific. We found that ENaC expression was the same in wild-type and Pendrin knockout mice, and in the thyroid and in the colon. And interestingly, if you knock out Pendrin in the inner ear, ENaC abundance and function actually goes up. So it goes in the opposite direction of what you see in kidney. So how might Pendrin modulate ENaC abundance and function? Well, it may do so through a protein or through a cell-cell interaction. It may do so through the release of a second messenger. Or finally, it may do so through changes in the downstream composition of the luminal fluid. So there are a couple different models that have data to support this. And in particular, our group has found that Pendrin mediates bicarbonate secretion, and that in turn, increasing luminal bicarbonate secretion stimulates ENaC subunit abundance and also ENaC function, including ENaC open probability. So the way that the model that our data support is that Pendrin mediates apical chloride bicarbonate exchange, which raises the bicarbonate concentration in the downstream luminal fluid. That then stimulates ENaC open probability as well as ENaC subunit abundance in the principal cells, which results in increased sodium absorption in the principal cells within the cortical collecting node. However, Pendrin gene ablation may also alter ENaC abundance and function through changes in an intermediary transporter. So, for example, Young-Hee Kim has shown that if you knock out Pendrin, you greatly downregulate the expression of the proton ATPase, which is expressed in the basolateral region of the type B intercalated cells. And of course, one would predict that with downregulation of the proton pump, you would have accumulation of ATP. And of course, ATP is a powerful signaling molecule that greatly affects ENaC abundance and function. So Dominique Aladari's group has data to support a second model, and they hypothesized, based on their data, that Pendrin inhibits ATP-stimulated prostaglandin release into the luminal fluid, which then stimulates ENaC subunit abundance and function. And their model goes like this. With Pendrin gene ablation, you greatly downregulate the proton ATPase. That then results in the secretion of ATP into the luminal fluid. ATP then binds to purinergic receptors along the apical plasma membrane of both principal cells and also type B intercalated cells. That then stimulates the release of calcium, which increases the release of prostaglandin E2. Prostaglandin E2 then acts through a receptor-mediated process to inhibit ENaC abundance and function. So the key points are that aldosterone regulates Pendrin, which likely contributes to the pressure response of this hormone. Pendrin stimulates ENaC abundance and function in part from the increased luminal bicarbonate concentration and by suppressing ATP-stimulated prostaglandin release. So in the last series of the studies, we have asked whether or not the kidney is the sole cause of the lower blood pressure that we see with Pendrin gene ablation. Well, as you know, blood pressure is regulated not only by kidney, but also by the brain, by the adrenal glands, and the vascular tissue. So we asked whether or not Pendrin might modulate blood pressure through its activity in some of these other tissues. Well, we've not found Pendrin expression in the brain, although that doesn't eliminate the possibility that it's there. But more recently, we've looked at the adrenal glands and vascular tissue. So this is a immunohistochemistry that young He Kim did of adrenal glands from wild-type mice and Pendrin knockout mice that were labeled as Pendrin. And as you can see, Pendrin is actually expressed in the adrenal medulla. This labeling is not seen in adrenal glands from Pendrin null mice. So then we asked if Pendrin might modulate adrenal function, and this data was presented by Yoscali, Lazar, Fernandez at this meeting. And so what Yoscali did was to implant chronic indwelling catheters in the jugular vein of mice. And then he let the mice recover for about 3 days. And then he measured blood pressure and circulating catecholamine concentration under basal conditions. Yoscali then used a model of acute stress by taping the mouse to a board for 20 minutes and then repeating the blood pressure and the catecholamine measurement. And this is what he found. Under baseline conditions, circulating epinephrine concentration, which is mainly released by the adrenal medulla, is really similar in wild-type and Pendrin knockout mice. Following the administration of the mobilization stress, of course, epinephrine goes up dramatically in wild-type and in Pendrin knockout mice, although it seems to go up by about 50% more in the Pendrin null relative to the wild-type mice. There may also be an increment in the release of norepinephrine in response to stress in the Pendrin null mice. We're currently investigating that. So then Yoscali and Roy Setliff looked at blood pressure by telemetry in both groups of mice following the same treatment model. And they found that under basal conditions, blood pressure is about 10 millimeters of mercury lower in the Pendrin knockout mice, which are shown in red, relative to the wild-type mice, which are shown in black. Following the mobilization stress, blood pressure increases by about 10 millimeters of mercury in both groups. But the difference is that following relief of the stress, the blood pressure returns to baseline levels within about 20 minutes in the wild-type mice, but remains elevated for longer in the Pendrin knockout mice. So Pendrin gene ablation appears to alter the pressor response to immobilization stress. And finally, we looked at Pendrin expression and its effect on vascular tissues. So in collaboration with Roy Setliff, we looked at the mouse aorta. Well, these are Pendrin expression in lysate from kidney and also mouse aorta that were taken from wild-type and Pendrin knockout mice. And you can see that while Pendrin is robustly expressed in kidney, we didn't see any Pendrin expression in the aorta or other vascular tissues. But nevertheless, Pendrin gene ablation seems to markedly change aortic contractile force. Roy Setliff has observed that in response to either phenylephrine or angiotensin II, the force per cross-sectional area of the aorta is markedly increased with Pendrin gene ablation. So, in conclusion, Pendrin gene ablation produces a natriuresis and a chloreuresis, which reduces blood pressure. However, it also increases stress-induced release of catecholamines from the adrenal gland, and it also appears to increase vascular sensitivity to catecholamines and angiotensin II. Pendrin and ENaC interact. Pendrin, as I mentioned, is a chloride bicarbonate exchanger expressed in Piscean intercalated cells that mediates bicarbonate secretion and chloride absorption. In response to aldosterone, it's upregulated, which increases the bicarbonate concentration in the luminal fluid that then stimulates ENaC, both in terms of open probability and also channel subunit abundance, which increases sodium absorption. However, the second mechanism, which is supported by the data of Dominique Aldosterone's group in France, is that following aldosterone administration, Pendrin, of course, is upregulated. That then stimulates the proton ATPase on the basolateral plasma membrane. That reduces ATP secretion into the luminal fluid. And the fall in ATP concentration then stimulates the epithelial sodium channel. And the result is increased sodium absorption along the cortical collecting. So aldosterone stimulates sodium chloride absorption in part through ENaC and Pendrin mediated sodium chloride absorption, and the result is an enhanced pressure response to aldosterone.
We'd like to invite you all to join us at the banquet tonight, where we're going to present the awards to Susan, and then congratulate her again on a fabulous tour.